Here's what Harold Katcher's plasma fraction is. Harold Katcher plasma fraction is a combination of small extracellular particles isolated from pig's blood. Okay, thank you for watching. Bye! No, I'm kidding. In this video, I will explain how I know that, what it means, and new developments in this research line. First of all, my name is Nina, and I'm the Vice President of the Rejuvenation Science Institute. I'll talk more about this institute and our work later in the video. But first of all, who is Harold Katcher, and why would you be interested in him or his work? So Harold Katcher is a researcher from the United States who was involved in the discovery of the human breast cancer gene in the 1990s. He is a biologist, and after working some years in research, he started working in teaching in the University of Maryland. Later in life, he discovered the work of the Conboy lab from Irina Conboy and Michael Conboy, and their research was about plasma dilution and heterochronic parabiosis. They were researching these two subjects to understand why, when you put old blood into young rats, the rats appear to age. So the Conboys discovered that when you dilute the blood of old animals, they actually kind of rejuvenate. They seem healthier and more active. So Harold Katcher read the papers from the Conboys lab, and he created a hypothesis that maybe there are pro-aging factors in old blood and pro-youth factors in young blood. So the idea is that the age of the animal is controlled by signals in the blood, and there are signals that indicate that the body should be old and signals that indicate that the body should be young. So he proposed that if you put pro-youth signals into an old animal, the animal would actually rejuvenate. So that is precisely what Harold did. So in 2020, it was published an article in bioRxiv, which is not a peer-reviewed journal, it's a repository for articles. He published an article describing that he isolated from young pig's blood a plasma fraction that, when injected into old rats, was able to rejuvenate these old rats, including by epigenetic age measurements. So the rejuvenation that they reported in this paper was of 54% of epigenetic rejuvenation. But they also looked at other factors like grip strength and memory and several blood markers. But the problem is that in this 2020 bioRxiv paper, Harold and his team didn't explain what the plasma fraction was. So we actually had to wait like 3 years until 2023, when in October there was a new paper from Harold and basically the same researchers, in which they, in Geroscience, which is a peer-reviewed journal, in which they described more precisely what this plasma fraction is. So in this 2023 paper from Geroscience, they basically described the isolation of small extracellular particles. Small extracellular particles are nano-sized particles that are present in the blood and in other fluids from the body that include small extracellular vesicles and other particles. In this paper, besides describing more precisely how they did the isolation, they also did a test called the Western blot that showed the presence of some proteins that confirm that this plasma fraction contains small extracellular vesicles. And small extracellular vesicles is something that the cells secrete and are involved in intercellular communication. So they are being studied nowadays as the possible mechanism in this pro-youth factors idea. So there are several other studies from other scientists from around the world. When they inject small extracellular vesicles that were isolated from fluids in the body of young animals or even from the extracellular matrix of stem cell cultures, and they inject these young small extracellular vesicles into old animals, and they observe rejuvenation and improvement in health and even an increase in longevity. So there's an important distinction that I have to make, that Harold didn't confirm that his plasma fraction is only small extracellular vesicles. He confirmed that small extracellular vesicles are present in his plasma fraction, but he didn't do a more extensive characterization study to see if there were other things in the mixture. That's why we have to use the more generic term small extracellular particles, that it's actually what the International Society of Extracellular Vesicles recommend. So all this information is from the 2023 Geroscience paper by Harold Katcher and other researchers, and also some things that I talked about is based on other work by other scientists that will be linked here in the video. But one important distinction from the 2023 paper and the 2020 paper is that Harold and his team did a new epigenetic age measurement, and now they reported that actually it was a 67% epigenetic rejuvenation in the old rats. So this created a lot of buzz in the rejuvenation and the longevity space. But a lot of researchers are a bit puzzled, like, why is this so effective? Because this is the most impressive rejuvenation results in terms of epigenetic age ever reported in mammals. So this is one important reason why this experiment, this Harold Katcher experiment, needs to be reproduced by third parties. So other scientists in other parts of the world need to analyze the published paper and try to reproduce it. And that's exactly what the Rejuvenation Science Institute, which is a non-profit research institute based in Brazil that was co-founded by me and my husband Nicholas, is trying to do. So in 2024, we already actually isolated the plasma fraction exactly as it is described in the paper and injected in the same amount described into young rats just to check if there was any kind of acute immunological or toxic reaction, because the amount Harold reports that was injected is a very big amount. And now we are preparing to do a second experiment that will be a rejuvenation experiment. So we will have very old rats, the same type of rats that Harold used, that are called Sprague-Dawley rats, and we'll have them in the same age, which is 25 months, which is very old for a rat, because a rat usually lives at most 3 years. And we will do again exactly the same protocol. We will isolate the plasma fraction, and now we will inject it into old rats. And this experiment will happen in 2026, in June of 2026, basically because we have to wait for the rats to age. Another thing is that if the rats show signs of rejuvenation, we want to keep the rats alive. We want to keep injecting the plasma fraction into the rats and see how long they will live. So if Harold's theory is correct, if the plasma of the young pigs actually have pro-U factors, in theory, the rats could live forever. But to do all of this work, we need donations. We are a non-profit research institute that we are doing a partnership with the University of Campinas, Unicamp, which is based here in Brazil, in the same city that we are based, and is basically one of the best universities in Latin America. Our partner is Professor Marcelo Mori. He's world-renowned. He publishes in Nature, in Science, in very important research magazines, and he has a lot of experience with small extracellular vesicles, besides having a very well-equipped laboratory. So right now we are raising funds to do this 2026 experiment. We need a total of $75,000, and right now we have about 72% of that, like 50-something thousand dollars. We are also doing this in partnership with several donors, including DoNotAge.org and Healis, which is a NGO from Belgium. And if you want to see this kind of research to move forward, if you want to see if Harold Katcher was actually right, if his experiment is reproducible, I invite you to donate to our institute. I'll leave the links below. Thank you for watching. Bye bye.